to welcome you all to our first series of C.R.E.A.T.E, which is a learning initiative by the Emirates Diabetes Endocrine Society and Sharjah University. And it's given me a great opportunity. Probably you all know me. I'm C.R.E.A.T.E Al-Awadi. I'm president of Emirates Diabetes Endocrine Society and a consultant endocrinologist and head of endocrine section at Dubai Hospital. And it's given me a great pleasure, during this webinar to take this opportunity on the behalf of Emirates Diabetes Endocrine Society Board to thank Sharjah University for this great initiative, as we had collaborated with them. So thank you to all the administrative at Sharjah University, to all their faculty, to all the staff who had made this possible. And special thank to Professor Bashair, who was the dynamo behind this initiative C.R.E.A.T.E, which Dr. Bashair will tell you in detail what does it mean. So before we start, I just want to remind you that you can connect through also our live stream, through our EDS YouTube channel, and also the webinar will be available on demand through our EDS Institute. And during the course of webinar, if you have any question, please feel free to type your question into the chat box, which then at the end of the talk of our speaker, Professor Bashair, will be taking this Q&A. So please make sure to ask many questions to make it interactive. And I just want to remind you that this webinar series will be CME accredited by the Dubai Health Authority, but please, all participants need to, fill the survey post the webinar in order to obtain your CMEs. And now I would like to, present Professor Bashair. As I mentioned, Bashair was behind this series of webinars. So Bashair is the program director of this C.R.E.A.T.E. She's an associated professor at College of Medicine, and she's the deputy director at University Sharjah Diabetic Center and the coordinator for the MSc in Diabetes Management. It's a pleasure to have you, Professor Bashair, and the floor is yours. Thank you.
Thank you very much, Dr. C.R.E.A.T.E, for the introduction and for giving us a great opportunity to share our initiative and to make our learning processes very rich and to share our expertise and our knowledge with our colleagues in diverse communities, either medical communities or scientific communities. I have only 2 slides, just trying to move to the next slide. I have 2 slides in which I just want to introduce the initiative, and after that, I will be very happy to introduce our honorary speaker for today, Professor Rory McCrimmon. So I think for audience like you, I shouldn't spend a lot of time talking about our global health challenges and how today, looking at what we're experiencing today, is the list of these challenges is increasing and the nature of it is becoming very complex and interconnected. So I think it is very important whenever we think about these challenges, we think about the stakeholders in terms of organization, of institutes, of different entities who's trying to overcome these challenges. And to be more relevant to what we're talking about today, it is very important to mention the contribution, the significant contribution of research. So research being one of the historical and fundamental processes in helping us to overcome all the health problems and health challenges that we are facing. And it is part of our history, it's part of the present, and it is shaping the futuristic innovative solutions. So looking at the group of people who are looking after this process, researchers, they are naturally gifted and talented. They do have a special set of skills helping them, enable them to identify the problems and design customized protocols to find the right solutions. And although we do have a lot of them in the world with different type of researchers that they're doing, but we still, I think we need to collaborate together and put our efforts together as researchers so that we can come up with impactful solution for these challenges. And like was one of the reason that we established the initiative C.R.E.A.T.E in 2018. At that time, our institute, and to be more specific, I'm talking about the medical campus in our university of Sharjah. The strategy at that time was to enhance research activities and research productivity. So at that time, we said the best way is to put our collaboration together, our effort together, and leverage the expertise, the skills, and the knowledge of our researchers, and start this process of continuous research education. It was a very successful initiative, and many groups within the academic teams learned and improved based on that and move forward on that in the field of doing research. However, it's very important to remember that when we've done that, we've done it within this kind of internal community. And most of the time, because the academic teams do the research internally, it is highly associated with more basic research. I'm not talking about 100%, of course, there is some people doing translational research, but the majority is involved in basic research. So we thought that to step out of that and to look at broader kind of community and to recruit more people with a different sets of expertise, knowledge, and experiences, it's better we really kind of talk to our colleague from the other side, which is the clinical teams. And it's very important to mention that we have an excellent group of clinician and clinical teams who are doing a great job when it comes to practice. But the communication and collaboration between the teams, in academic teams or basic researchers and clinical researchers, it's not in the right place. So we saw that, what about we collaborate together, we exchange our expertise, our knowledge, and we improve the outcome. So by working together, we'll definitely have more impactful outcomes. At the end, we'll kind of meet the ultimate goal that all the research is working for, which is the patient's health and quality of life, improving their quality of life. So this is led to C.R.E.A.T.E 2025. Today we have our first day, and I would like to thank all the people who are here today to help in implementing this initiative and make the outcome of it is more significant. So what we are aiming, the general aim is to bridge the gap between research and clinical practice by sharing the same platform, sharing our knowledge, sharing our challenges together, and learn together in one setting. So just quickly, I will go through the map of the program. I will just show you what we are trying to do. It is a long-term initiative, so we're looking at 6 to 12 or even more months. And here is the overview of the program. I'm trying to go to the next slide. I don't know why it is not working. Just I think it is frozen. Yeah, okay, great. So here is the overview of the program. In the first stage, or we call it first phase, we would like to focus on awareness and training. But we know that there is a significant number of clinicians who are already doing amazing research, but we want to start with, the majority of the clinical teams who did not have the chance to know more about the significance of research and how it is very important to involve in research. So we will have the awareness that will be a set of webinars. Today we're starting the first one, and it is for healthcare professional, but for everybody else who's interested in doing research. After that, we'll have the training where we will have a series of workshops to give more kind of hands-on experience for the people, and will help people to start thinking and initiating their proposal. The second stage after that, we are establishing a support system. We'll try our best to establish a well-structured and immediate kind of communication between researchers to support their processes. So we will have the support along with our training processes. And in the fourth part of the second stage, we are emphasizing of the fact that without increasing the productivity of our researchers, clinicians, and scientists, it won't be having that, like kind of significant meaning when it comes to what we're doing now. So as we all know, research without outcomes, without publication, wouldn't really have the same impact and have this kind of broad impact. At the end, the third phase, and to be honest, this is, like a suggestion from Dr. Fathia, we would like to establish UAE research network. So from today, whoever is participating in this program, in this initiative, by default, they are part of this network, research network. What is the main aim of it? It will be a platform for continuous training and education, and on the same time, establishing large groups to kind of run big studies at the level of the country, or even at the level of the region. I'm done here. Thank you very much for your attention. And now we'll move. It gives me a huge, actually, pleasure to introduce Prof. Rory McCorman. And I should say that I met him face to face in 2022 when we organized a symposium about advances in neuroendocrine and metabolic access. It was a great collaboration, and since then I'm just enjoying collaborating with him. Prof. Rory, he's a professor of experimental diabetes and metabolism. He's the Dean of the School of Medicine, University of Dundee, Scotland. He's recognized for his pioneering work in understanding how the brain responds to hypoglycemia and its implication in type 1 diabetes. As a clinician and researcher, his work led to groundbreaking investigations into the neurobiology of hypoglycemia. He involved in identifying key pathways such as activated protein kinase, ATP-sensitive potassium channels in glucose-sensitive processes. His research has been highly influential, and that's very clear, contributing to improving clinical understanding and treatment strategies for diabetes. He published a huge amount of work with 20,000 citations, reflecting how impactful his work is in the field of diabetes. He received multiple prestigious awards, including Dorsey, Hodgkin, and R.D. Lawrence Lectures from Diabetes UK, and election to Royal Society of Edinburgh. It is a great today to listen to you, Prof. Rory. We are waiting to listen to you when you share with us your point of view about translating science into care, the importance of clinician scientists. The floor is yours, and thank you for your time and for being here.
Thank you very much for that kind introduction, Professor Bashir, and thank you also to Professor Fathia for inviting me to give this talk, and welcome to all of you who have joined this webinar online. It is a real privilege of me to be here and to talk about the importance of translating science into care. Although, of course, Dr. Bashir has beautifully illustrated quite how important that role of the clinical scientist is. And I might extend that into the clinical academic as well as the clinician, because I think all of us involved in clinical practice or research or teaching have multiple roles in the course of our careers. And I will perhaps illustrate it through my own career and some of the work I've done. And I hope you don't feel that is too egotistical, but perhaps it's an example of how careers are often not straightforward. They're often have different trajectories, and sort of illustrates the type of work that we do. You can hear me clearly enough, I hope, Professor Bashir, yes?
Yes.
Great.
Yes.
So if I move these slides on, so this is my academic career. I'm getting old now, so I've been around for a while. And I, graduated and met from medical school, as many people, of course, all of us will do. And then I went through a clinical training. And my first step in my research career came when I did my medical doctorate with Brian Freer in Edinburgh in the 1990s. And in truth, the reason I did a medical doctorate was at the time, you could not get into specialty training without a period of research in the UK. It was a bottleneck. It was very competitive. So I had no particular intention of becoming a clinical academic, but felt that I had to do that. And I did that period. I enjoyed that period of work before then going back into clinical training for my specialty training. But having enjoyed research and having an opportunity during my specialty training to take more time out, I then took a postdoctoral fellowship at Yale University in America to do a different type of research. And I thoroughly enjoyed that as well. I still came back. I became an NHS consultant physician in Liverpool as a general physician with an interest in diabetes and endocrinology, but decided that wasn't the right career for me. And I resigned from the NHS and went back into the university, back to America, where I eventually established my own lab and had funding from NIH, and then transitioned back to Scotland, again at a later stage, to continue my work in research initially, and increasingly with an academic role of ultimately ending up as the Dean of Medicine here in Dundee, but continuing my research program, and obviously continuing my clinical work. I still do clinics and I still do on-call because I think core to what I am is a doctor. So hence that illustration for the winding road, because for many of us, we come in and out of this at different times, but I think most medics, in my experience, have an awful lot to offer and can think of different ways in which they can both research or innovate or change care pathways and take part in commercial trials, academic trials, all of which I think are very, very helpful to advancing care and addressing some of these global challenges that we have, both within healthcare systems and society. Now, during this period, the type of research I've done is varied. In my initial work in Edinburgh involved human studies, experimental medicine work. When I went to America, I focused more on animal studies because I was interested in basic mechanism. I continued that work when I came back to Dundee, where I focused both on animal studies as well as human work, and also started to do a little bit of cell culture work, and then increasingly moved between the bench and the lab in pretty much everything that I did. And I'll illustrate that through some of the work. So the first phase of my journey now, this was in Edinburgh when I did my Doctor of Medicine degree. And that focused really on the impact of low blood glucose on cognition, because hypoglycemia was very topical at the time. There was a lot of interest in this field because insulin therapy had come in. People were now thinking about more intensive insulin therapy management, and that was leading to a lot of people experiencing low glucose. There were questions being asked, both in the transition from animal to human insulin, and whether that affected symptoms and cognition, and just more evidence of people developing cognitive impairment during low glucose. So I started to look at that, and I was interested in a task called the information processing, which really relates to brain power, that factor G that underlies our basic processing speed, which is a sort of marker of intelligence, and looked at the impact of low glucose on that. I learned how to do a hypoglycemic clamp study. I learned about experimental design. I learned about statistical analysis. I learned about patient recruitment and working with people and getting them to take part in clinical trials. Incredibly valuable part of my experience. I also had 2 or 3 years out where I was steeped in my speciality. I was learning about diabetes in real depth in a way that sometimes is challenging to do on the clinical floor. And it ended up in a paper in Brain. I was delighted. I'm always very proud that I ended up having a publication in Brain, which sounds much more impressive than it probably actually is, but it was good at the time. My second phase of the journey was actually to move back to the United States, and as I talked to you before, and I was now interested a little bit more in mechanism. I enjoyed my work within cognition, but I decided that what really grabbed me was trying to understand those basic mechanisms that led to the hypoglycemia counter-regulatory response. Why is it that people responded to hypoglycemia the way they do, and why were they more prone to severe hypoglycemia over time? Now, the reason that this was considered important largely came from the results of the Diabetes Care and Complications trial. Many of you, if any of you have been around as long as I have, know that this was a very seminal study published in the New England Journal in 1997. Well, this is 1993, actually. The hypoglycemia data was published in 1997. And what this trial showed definitively was that intensive glucose lowering, aiming to near normalize glucose control, led to a marked 75% reduction of complications with this sigmoidal relationship between HbA1c and each microvascular complications, be it retinopathy or any form of microvascular disease. And of course, we learned through C.R.E.A.T.E both macrovascular disease as well. But at the same time, as HbA1c dropped, people started to experience severe hypoglycemia, to the extent by the end of the trial, almost every subject had had a severe hypoglycemic episode. And this was both distressing for the individual and for their carers. And people were experiencing a lack of awareness of those hypoglycemic episodes. So I went to work with a chap called Bob Sherwin, who I'll introduce to you in a second, at Yale University. It's my first postdoctoral fellowship. And I looked at this idea of, well, how did the brain sense a low glucose? Because the brain was considered to be fairly central to this whole process. And there was some evidence that this might occur in the brain from some earlier studies. So what you have here in the center is you've got the pancreas, and it's a classic islet with beta cell producing insulin and the alpha cell producing glucagon. And we know about the importance of glucokinase, the ATP sensor potassium channel within the beta cell, etc. There's been lots of research in this area. On your left-hand side, you'll see a picture of the fly brain. And in the fly, insulin and glucagon are actually neurotransmitters found within the brain. So the work I pursued was on the right-hand side. I was looking at a particular part of the hypothalamus called the ventromedial hypothalamus. And I was looking at this idea that we had the equivalent of the beta cell, the glucose-excited neuron. So this is a neuron whose firing rate increased when your glucose levels were high. And the glucose-inhibited neuron, and that's a neuron a bit like the alpha cell, whose glucose levels, whose activity increases when glucose levels fall. Now, this work, as I said, had been started out by Bob Sherwin, who was a wonderful American clinical academic who did had many contributions, including the insulin pump, some of the big things that he drove that affected type 1 therapy. And Bob Sherwin had shown a number of years previously that if you microinjected to this special area of the brain, the ventromedial hypothalamus, something called 2-deoxyglucose, which is a non-metabolizable form of glucose, so it creates local hypoglycemia, well, that initiated a counter-regulatory response. He also did probably the more important experiment, was that he created, using that same clamp technique that we used in humans, in animals, he created systemic hypoglycemia, but at the same time infused glucose into this tiny, tiny area of the brain in the animal model in the rat. And that suppressed counter-regulation. So that provided really good evidence that the brain was very important. And this particular part of the brain was really important in how we sense glucose. So I went there really to start to think about, well, what were the different potential mechanisms? And I looked at a number of different areas. Professor Bécher kindly highlighted the work on AMP kinase. And there was a series of publications that I did at the time, really quite a number, both looking at manipulating AMP kinase within the ventromedial hypothalamus, as well as looking at the more systemic effects of AMP kinase to show that it was really important in terms of how blood glucose was regulated during hypoglycemia. And here is just an example showing that effectively, if you activate AMP kinase in the brain during systemic hypoglycemia, you could drive the counter-regulatory response. So you can see, compared to the control studies, in green, you can see much bigger responses of glucagon during the hypoglycemic challenge. And this was also interesting at the time because people had not been able to show this brain regulation of glucagon secretion. It was presumed to take place almost entirely within the pancreas. And here we're showing that actually the brain also has a potential role in this multifaceted regulation of the alpha cell. We also showed that if you knock down AMP kinase in the brain using viral vectors, you could suppress the counter-regulatory response, you could deliver AMPK activators, and you could activate the response. So through lots of different techniques, we showed that this system was important. And AMP kinase was, at the time, a chap called Graham Hardy had done a lot of work to show that this was actually a key metabolic fuel sensor, broadly within different cell types. And Graham's work has now been cited many, many, many times. And AMP kinase is now recognized as one of these really critical enzymes in all cell systems. Now, I also went to take on these techniques, and this illustrates a little bit more of the sort of techniques that I use. I use them in multiple different ways and in lots of different approaches. So on the right-hand side, you'll see this, what's called the clamp technique. So in the clamp technique, whether you're using an animal model or in human, you infuse quite high doses of insulin. So you have a constant high dose of insulin. That will tend to make the glucose full. You infuse dextrose there, and you infuse dextrose at a variable rate. So you can fix glucose at either euglycemic levels, say 5 millimole, or different degrees of hypoglycemia, 3.2, 2.8 millimoles per liter. And you can keep the glucose level fixed at that. And you can compare different hypoglycemic exposures between different, under different conditions. On the left-hand side, you'll see what's called the microinjection technique, where animals undergo surgery and they have cannulas implanted that target different parts of the brain. And then you can, when the animals are awake and moving around, you can inject your compound, your pharmacological compound, or your viral vector, then you can assess the impact. Now, I was therefore looking at the role of the C.R.E.A.T.E channel, and what we showed, and here in this first slide, if you microinjected the C.R.E.A.T.E channel blocker, glibenclamide, directly into the VMH during systemic hypoglycemia, you suppressed, shown here in green compared to the control study, the epinephrine, the counter-regulatory response to hypoglycemia. We showed on the other side that if you inject a potassium channel opener diazoxide or a C.R.E.A.1 selective potassium channel opener NN414, you could conversely augment, amplify the counter-regulatory response to hypoglycemia, and this showed a potential role of the C.R.E.A.T.E channels, again, in glucose sensing. And we did a number of other subsequent studies leading on to a human study that I'll present to you later on that showed actually this might be a viable treatment for people in the future. So my third phase of my journey then, having completed a lot of these basic studies, I worked at AMP kinase, I worked on the C.R.E.A.1 C.R.E.A.T.E channel, I worked on the corticotrophin releasing hormone system, and the neurotransmitters glutamate and GABA, so a whole series of work looking at what these fundamental sensing mechanisms were. And that was great, and I was really interested, but I'm a doctor, and what a doctor wants to do is to come up with cures and to figure ways of treating people. So when I came back to Dundee, I was really keen now to move on to that next phase where I could start to introduce things into clinical practice and start to have an impact upon the disease itself. So I did a couple of things. One was to look further into mechanisms, and I now started to look, I realized that if you wanted to look at signaling, you had to work within cell culture systems. And I'm illustrating this because it was both a lesson for me, and it was both valuable, but also a lesson for me. It was valuable because I learned a lot about signaling, but also valuable because I realized I really didn't want to learn a lot about signaling, that I was too much of a clinician, and I wanted to move, actually, I learned to move away from that system and to leave that to scientists so I could get on doing with more of the clinical work. But I was looking at a very specialized population of neurons, hypothalamic glucose sensing cells, and it did teach me a number of things. So in the first slide here, you can see these what are called GT17 cells, and these are gonadotrophin releasing hormone cells. And you can see that when we have these glucose and glucose 2.5, this is in this top chart, these cells are firing away. And if we drop the glucose to 0.5, they hyperpolarize, and they put glucose back in, they start firing away. And you can show a linear relationship depending on the glucose level to which they're exposed. Now, in a very similar level, the beta cell, you can also record from. In many ways, what's interesting is the beta cell is like a modified neuron. It's very intriguing. It's one of the only systems outside of the brain that you can record from electrophysiologically. And if you expose the glucose to 10 millimole glucose, they're firing away nicely. You drop the glucose to 2, they hyperpolarize, you increase the glucose again. So you can show that this is, the GT17 cell is just like a beta cell. The differences you will notice are in the glucose concentrations. So a thing that we learned very quickly is that the glucose concentration in the brain are about 20% of those in the plasma. Really interesting that it's far, far, far lower, the glucose levels in the brain, and how you need to study cell systems. So here we had a good model system, and we went on to show that if you gave a glucokinase activator here, you could actually drive a firing rate even though the glucose level was low, that you could, during a normal glucose level, you could use a C.R.E.A.T.E selective K2P channel blocker and block it, and you could knock out AMP kinase and you would lose glucose sensing in those cells. So again, this cell, very much like the beta cell, very much like the work I'd done in the brain, used those classical hallmarks of a glucose sensing system, glucokinase, AMP kinase, and SIR1 K2P channels in order to take that glucose signal, that change in glucose, and translate it into an output, a change in action potential, just as the beta cell does the same thing with glucose and changes it into insulin secretion. So that gave us a model system that we could actually work with. And I'll illustrate that a little bit later on. It also showed to me and reminded me of the importance of the K2P channel. And as I said to you earlier, I was keen to come back to the UK, start to think about clinical translation. So what I did in this study with a clinical research fellow working with me at the time, Priya George, was to take people with long-standing type 1 diabetes who had hypoglycemia unawareness, perform those glucose clamps, and here you have someone with type 1 diabetes with what's called a hot box, where we arterialize the blood glucose, we make it more like arterial blood, where we measure it every 10 to 15 minutes. And here we're infusing insulin and glucose, trying to manage the blood glucose level. We're sampling regularly for hormones. We're doing symptom and cognitive function tests over here to look at how people respond during the low glucose. And we gave the non-selective potassium channel opener diazoxide 30 minutes prior or 60 minutes prior to the hypoglycemic clamp. And what we showed was a 40% increase in the epinephrine response and an improvement of symptoms. So that was fantastic. So now we would show that in clinical practice, the work that we had done within the cell and within the animal translated into the human were a very, very similar effect of potassium channel openers. Now, a further lesson here is how you then work with industry, because I was going to work with Novo Nordisk with their SIR1 selective ATP sensitive potassium channel opener, which I thought would be a very specific treatment and very good for people with type 1. And they did, but they wanted to use it for people with type 2 diabetes. And in a clinical trial, there was some abnormal liver blood tests, so they pulled it from the market. So I was no longer able to work within that space. And that, I guess, I'm afraid, sometimes where you think you're having a very successful journey, it can, for reasons beyond your control, suddenly stop being an area where you can pursue further. But again, this showed the importance of translational work, thinking again about lab, bench, human, and what we can do to make that transition. And this was early days of pharmacogenomics as well. We actually showed that if you looked at this thing called the KCNJ11 mutation, which affects the shape of the K2P channel, you could actually look at how people responded to diazoxide and their counter-regular response, even in a small group of 12 subjects. And I think that's what we're beginning to see, as many of you will know, in clinical practice, where I think pharmacogenomics are going to really have a huge effect upon clinical practice in the future. I mean, it is now £40 to get your genes sequenced. And I think what we're beginning to see is the fact that people will have their genes sequenced, and we will be looking at how this influences our prescribing in the community. Now, the next thing that I wanted to do is to understand this concept of hypoglycemia unawareness. And as many of you will know, that people with long-standing type 1 diabetes, and particularly those who are exposed to recurrent hypoglycemia, develop reduced awareness. You can take someone without diabetes, you can drop their glucose to 2.5, and they'll tell you about it. They will be pale, they'll be sweaty, they'll have increased heart rate. But if you take someone with long-standing type 1, particularly someone who's had lots of hypoglycemia, and you drop the glucose for 2.5, you'll get no response whatsoever. That patient will be sitting there quite comfortably, feeling quite well and unaware that something is happening. And of course, the great danger there is that your ability to respond to a further drop of glucose is impaired. And once the blood glucose is down close to 2, you're going to have a severe hypoglycemic episode, and there's not a lot that you can do about it. Now, some of this work was beautifully illustrated by work that Simon Heller did when he was working with Phil Cryer in St. Louis. And he took subjects into the lab that on day 1, they underwent 2 hypoglycemic episodes, and then all control studies at euglycemia. And then the second day, they were made hypoglycemic again. So you can see in a sort of purpley color here that on the first hypoglycemic episode, they have this very brisk epinephrine, adrenaline response to hypoglycemia. But the second day, after 2 episodes, you now have this marked reduction in the exposure to hypoglycemia. And that was really interesting. Phil C.R.E.A.T.E then came up with the concept of hypoglycemia begets hypoglycemia and called this a maladaptive response. Now, I tended to disagree with that term, and for reasons that I'll explain to you in a second. And that's illustrated a little bit on this slide on the right. So many of you know, who look after people with diabetes, will see people with long-standing, particularly type 2 diabetes, and often with type 1, who will tell you they start to feel unwell, even though their glucose levels are 6 or 7. And this slide illustrates it quite nicely. So here you can see the adrenaline epinephrine response to hypoglycemia in people without diabetes in the black circles down here. People with type 2 diabetes whose glycemic control is poor in the white circles at the top, and people with type 2 diabetes on insulin with a lower HbA1c down here. So you can see the group who are inclined to experience hypoglycemia have a slight suppression of the counterregulation. But the group who are exposed to chronic hyperglycemia, chronic high glucose levels, actually counterregulate at a much higher glucose level. And in fact, in clamp studies, they will have a hypoglycemic response, even if their glucose level is normal. And I think this suggested to me that we had these shifting thresholds. So what I mean by this is that as the blood glucose levels drop, it is not a linear relationship. What the body does is it recruits more and more of these glucose-inhibited neurons, and it switches off more and more of these glucose-excited neurons, and you get this gradually increasing counter-regulated response with a maximum effect at around about 2 millimoles per mole glucose. In people who have exposed to recurrent or chronic hypoglycemia, this system is shifted to the left. So your counter-regulated response is now occurring at a lower glucose level, even though your maximal response is the same. And in those exposed to chronic hyperglycemia, this whole system shifts to the right. And so you're now counter-regulating at high glucose levels, which is why I think we've got to recognize the need to lower glucose slowly in people with poor control, because it is very true that people, particularly with type 2 diabetes, will experience hypoglycemia at normal to slightly higher glucose levels, and they will start to look to eat food. And that, of course, makes the management very, very hard. Now, this important thing tells us that, and the other thing is that this is a universal phenomenon. I've showed you this can happen in cells. This happens within C. elegans, it happens in the fly, it happens in this thing called the sea slug, California aplasia, and I'll tell you about that in a second, but also in the mouse, the rat, and the dog, and the humans. So that suggests that this is a universal response, that almost all cellular organic systems respond to this type of challenge in the same way. And that's been quite important in my thinking as I've moved forward in this area. Now, what might therefore be the mechanisms for this? And there are lots of potential mechanisms. I was interested in the brain, so not surprisingly, I focused on this area and I looked at, conditions such as changes in glucose transport, changes in your ability to use alternate fuels, which I think is actually happening, particularly with type 1 diabetes, changing within those sensing systems I talked about, changing within neurotransmitter release or gliotransmitter release from astrocytes, or peripheral signals like opiates and steroids and cytokines, all of which have been implicated. And in truth, like any complex system, there probably are multiple mechanisms. There is no single mechanism that can be telling you what's happening. But I was thinking more and more about this adaptive response. And that's just finally to illustrate that you can do this in a cell. I showed you these these GT17 cells earlier on, and that the GT17 cells are firing away at a normal glucose, and then they hyperpolarize here with a low glucose. This is in the top bar. If you take those cells and expose them to low glucose in the petri dish on 3 separate days, returning them to a normal glucose in between time, and then study those same cells after 3 days, well, they no longer dissense glucose. You have an unaware neuron, and you can create this in a dish, and you can use that to look at mechanism. Again, highlighted the fact that this is a universal response. And I'll skip over this slide, because I think what I want to get on to is talk a little bit about a concept that then came up with called habituation. And this was looking at the idea that we actually have an adaptive system, that it's a system that learns how to respond to low glucose. And I think if you can picture this in your mind is that humans have not really been exposed to low glucose, insulin-induced hypoglycemia for many years. We've had insulin for just over 100 years, but we are a species that evolved over many, many years. What we have been exposed to is starvation. And hypoglycemia is an accelerated form of starvation. So how does the body adapt to and learn how to respond to low glucose? Well, in many ways, we learn how we adapt to many different stressful situations, including starvation. I'm not going to show some of the data. I can go on at length about hibernation models and different metabolic fuel flexibility and how the body does adapt centrally to low blood glucose challenges. But let's take the idea of stress. How does the body respond to repeated stress? And actually, this then took me back. This is the importance of reading widely and thinking differently, thinking outside of the box, to work that was done in the 1950s and the 1960s on this creature called the California sea slug, that is used for a lot of psychology and behavioral studies. And habituation is this concept where if you repeatedly stimulate, repeatedly stress this animal, and I'll show you how they do it in a second, you get a reduced psychological, behavioral, or physiological stimulus. What they do with this sea slug, and there are these old videos which are quite interesting to look at, is they flip it upside down, and it has what's called a friable mantle. And if you get a little wooden poker and you stick the sea slug with this poker, you get this withdrawal reflex, which is pretty much like a sensory motor reflex. And they use this to study this concept of habituation. So when you think of the reduction of psychological, well, that's reduced anxiety. And we see that with recurrent hypoglycemia. Reduced behavioral, reduced food-seeking behavior. Reduced physiological, well, we know there's an impaired counter-regulatory hormone as symptomatic response to hypoglycemia. So if you think of hypoglycemia as the stimulus, it actually sounds like it might be doing the same things as habituation. And that is what ultimately I set out to test. And this is the model. The model is, as I said to you, you've got this sensory motor reflex in the sea slug. Well, let's think of hypoglycemia now as your internally sensed stimulus and counter-regulation as your motor response. So you actually have the same system. Glucose goes in, there is a motor response coming out. And that has various systems, glucose sensing elements in the brain, there's premotor control, and there's an autonomic and neuroendocrine response. So I'm testing this sort of system. And what I'm going to do is I'm going to look at something called dishabituation. So dishabituation is when you introduce a novel stimulus, can you reset the system? As an example to you, anyone who's ever exercised or exercised regularly thinks that they're getting fitter all the time. And then they go and do something very different and suddenly think, oh my gosh, I'm exhausted. I've never been able to do that. And that fits with the idea of dishabituation. That if you come in with a novel stimulus, you reset that original response. So here you come in, you enter into these pathways and you stimulate again. So quite a hard concept to grasp, but here's how I tested it. So we took animals, in this case, rodents, rats, and we made them recurrently hypoglycemic. We had control animals, and here they're illustrated in white. We had recurrently hypoglycemic animals, some who've had a low intensity exercise, this is in purple, in orange, and some who had a single episode of high intensity exercise. So the animals were put on a treadmill, and that treadmill was gradually ramped up so that they were sprinting as fast as they possibly could. And they just had this one episode. And then the following day, they underwent a hypoglycemic clamp. So here are your control animals who are given saline for 4 weeks, 3 times a week for 4 weeks, and then are studied and made hypoglycemic. And they have this very brisk adrenaline response, just like humans do. Here in orange, we have animals who have 4 weeks of recurrent hypoglycemia, 3 times a week, so 12 episodes of hypoglycemia. Then they have their hypoglycemic clamp study. So the hypoglycemia is induced with a bolus of insulin, but then they have their clamp study where we fix the glucose level at 2.8 millimoles per liter glucose. And now they have this suppression of the adrenaline, which we've seen in multiple different ways. We see it in humans, we see it in rats and dogs, etc. In green, we have the rats who had 12 episodes of hypoglycemia. Then they had a single burst of high intensity exercise, and then they had a hypoglycemic clamp 24 hours later. And now they've restored their counter-regulatory response, both for adrenaline and glucagon. So that is consistent with the idea of dishabituation, that we have come in and we've introduced a novel stress, and we have been able to restore the response. We looked at mechanisms, as you're inclined to do now, using proteomics, and many of you who are involved these days in research will do some form of omics, trying to understand what might be going on. And again, I won't go into details, but suffice to show that what this suggested was that when there was recurrent hypoglycemia, you had reduced glutamate, so that's the excitatory neurotransmitter, you had reduced glutamate activity in the ventricular hypothalamus, and after dishabituation, these glutamate levels went back up again. So you're restoring this counter-regulatory response. Now, that's an animal model, it's not a human model. So again, it's a translational lab. So now we wanted to see whether the same thing occurred with humans. And so what we did was we took people with this, Katrina Farrell, who was my clinical research fellow at the time, took people with long-standing 24 years of type 1 diabetes, very good glycemic control, all of whom had impaired awareness. And we compared their glucagon, their responses to hypoglycemia after a single intervention. So this was on a treadmill. They had 3 episodes of 30 seconds sprinting with 2 minutes rest in between. And then the following day, 24 hours later, they had a hypoglycemic clamp technique. So these were long-standing type 1 diabetes with impaired awareness. And again, what we were able to show was that that single episode of high-intensity exercise meant that the following day, here in blue, their adrenaline and symptom score in response to hypoglycemia was improved. So what we have been able to show, at least in concept, was that dishabituation was also possible for people with type 1 diabetes. So again, it fits in with the idea that what we are looking for is a form of specialized form of learned memory called habituation, and that there may be ways without drugs that we might be able to change this practice. This was an acute episode. Clearly, this is something you want to be doing longer. So we followed this up with a study beautifully named HIT for Hypos, where people underwent 12 episodes of high-intensity exercise over a 4-week period, after which we did a hypoglycemic clamp, because we wanted to see, did this work in the long term, or was it just a one-off? And what we were able to show here in the dark blue compared to the control studies in light blue is after 4 weeks, there was improved adrenaline, there was improved noradrenaline, and for the first time ever in type 1 diabetes, we were able to show improved glucagon response to hypoglycemia. So 4 weeks of high-intensity exercise training was able to improve the counter-regulatory response to hypoglycemia in people with long-standing type 1 diabetes and impaired awareness. So again, this was a really exciting time for us to be able to show that we might make a difference. Just looking at this in a little bit more detail, there was better symptom scores in the group with high intensity exercise, both total and autonomic symptom scores consistent with our outcome. We use continuous glucose monitoring, as you would predict, and what we showed here, and I want you to look here at the blue bar, which is your high intensity exercise group compared to your control group in red. And what you can see, particularly overnight between 12 a.m. and 6 a.m. in the morning, there was less nocturnal hypoglycemia after the high intensity exercise, which again, against the whole idea that exercise makes you more sensitive to hypoglycemia at night. I think aerobic exercise does, but maybe anaerobic exercise doesn't. And this was much more pronounced. And here just showing you in details that the number of level 2 and level 1 hypoglycemic events, particularly overnight, were much lower after high intensity exercise. So there's an awful lot more for us to do within this space. There are a number of different criteria of habituation to really show that this is what is happening. But it then comes up with the possibility, can we do different things? We actually tried in animal models, we've shown that cold exposure does the same thing. So I'm very desperate to do the study where we have a sauna and maybe a cold pool afterwards for people to see whether that might make a difference. But I'd love to prescribe saunas and high intensity exercise for a period for people with type 1 diabetes to try and help them retain awareness of hypoglycemia, because of course there are lots of good vascular benefits as well. And I think it would be an interesting study to actually do. And that fits with this whole idea of impaired awareness and this idea of habituation, which we're driving, and looking at reasons why the body responds and changes the way it does. And part of me is just fascinated by mechanism, but a big part of me also wants to see how we can make a difference to people's lives. Now, very briefly, I want to also touch on what are the risks of hypoglycemia. I think there's been a lot of data recognizing that severe hypoglycemia is bad for us. Nobody wants to have a severe hypoglycemic episode. The experience is unpleasant, but actually there are long-term consequences of this. So this is work we did recently as part of a big consortium, and this showed in people with type 1 and type 2 diabetes that actually hypoglycemia is associated with an increased risk of cardiovascular disease, an increased risk of death, and even an increased risk of microvascular disease. And it's not occurring during the hypoglycemia, it's occurring over time. So why might this be happening? And here what I looked at was the idea that actually what happens is that you get oxidative damage and inflammation. So diabetes per se, high blood glucose levels, is a chronic inflammatory state, and that will deplete your antioxidant response over time. When you're hypoglycemic, and particularly in recovery from hypoglycemia, you see this profound oxidative stress. It's made worse when you go from euglycemia to hyperglycemia, because all that glucose pouring into the cells generates toxic free radicals. And I wanted to test the idea that this caused oxidative damage. Now, again, we were able to do this in clamp studies where we were able to modulate glucose. We were able to study animals with and without diabetes. We were able to fix their glucose, but also control their recovery. And what we were able to show in this study, and this is just looking at mechanism in an acute study, we were able to show that your body's ability to degrade damaged proteins was abnormal with recurrent hypoglycemia. And there was this reduced ability to repair proteins, which made you more sensitive to damage. We were able to go on and show that actually, if you look at something like cognition, in this STZ, which is a model of type 1 diabetes, there is impaired cognition over time just by having diabetes. But if you had diabetes plus hypoglycemia here in blue on the far right, you can see a real damage in terms of memory function within the brain. More recently, we've been looking at recurrent hypoglycemia in animals, looking at their cardiac function. And the paper that's about to come out, actually, in Diabetology has just been accepted. What we show is that actually recurrent hypoglycemia in this model of type 1 diabetes moves towards a dilated cardiomyopathy in an abnormal cardiac state. So hypoglycemia is bad for you. I mean, I think that's what it's saying, and something we need to look at ways in which we can avoid. Now, I'm aware we're running out of time here, so just a couple of editing slides just to show that, you know, obviously I work within that translational space, but I'm also a clinician. I'm involved really heavily in clinical trials, and I currently lead the diabetes research networks in Scotland, and in part of that is because we have these fantastic informatics systems. And I put this in because many of you are fully involved in clinical practice, and I think there's a lot of really good research that can be done in routine clinical practice, particularly if you're collecting good data. And we've got really good data in Scotland. We have 100% of our population on a database. We have linked data for their blood results, for their prescribing, for their hospital admissions, for their retinal screening, etc. And that enables us to do really strong epidemiological research on our population. We work in a collaborative way. If you scan Scottish Diabetes Research Network Epidemiology, if you scan that barcode, you can find over 80 publications now that have hugely influenced how we practice clinically. We also use this as a tool to recruit for commercial clinical trials, and we run a whole load where we're able to contact people directly to bring them into trials. We also run these large, so here is a project called iDiabetes we currently run in my region in Tayside, working with my colleague Ewan Pearson, who's really running this, where we've randomized our entire population of people with type 2 diabetes, nearly 3,000 people with diabetes, looking at how we can improve diabetes care, looking at novel biomarkers and using an electronic health informatics system. So broadly speaking here is that we now we look at apart from waist hip thigh ratios, ILF, we do things like what are called intelligent liver blood tests, which automatically push someone down the pathway through fiber scans. We look at NT-proBNP to look for heart failure, where we automatically cascade down towards echoes. We use type 1 genetic risk scores, antibodies, MODI testing to make sure we've got the right diagnosis. And then we can get people on the appropriate therapy for their conditions. So again, automated C.R.E.A.T.E, type 1 genetic risk scores, antibodies for diagnosis. We use the Exeter 5 drug model to give advice to primary care about what drug they should be prescribing based upon likely benefit in terms of HbA1c reduction. We more precisely use things like coronary artery disease, polygenic risk scores, and NT-proBNP to make sure we're identifying those with cardiac failure, making sure they're on SGLT2 inhibitors or GLP-1 receptor agonists. And again, try to identify those with fatty liver disease. So why are clinician scientists important? Well, I think, as Dr. Bashir said earlier on, Professor Bashir, it's important about translating research into practice and bridging that gap between the lab. But also there is direct clinical impact from what we do, both within the experimental medicine stage, but also in terms of innovation and changes to healthcare systems, projects such as iDiabetes. Driving a scientific process by saying, well, where are the gaps? Where are the questions that need to be answered? Fostering collaborations. You cannot do what you wish to do alone anymore. I need to work with basic scientists, but I also need to work with statisticians, epidemiologists, or people who understand health data, because I cannot be a master of everything. So team science is absolutely the future. Fostering collaboration is key. It is that unique experience. I think we scientists do not understand the clinic, and so it's incredibly important that we work with all of these specialty groups so they understand the clinical world that we work in and how a disease presents and how it progresses. And we are able to inform the research of agendas of our healthcare systems and our countries. And then finally, we have impact. What we do changes and impacts upon people's lives. So why is it important? As I said, we are dependent on top quality research. It's the only way that we can improve outcomes. And actually, it's the only way that healthcare systems in these days of rising costs can work innovatively. It should be core to everything we do within the health service. And there's a very clear correlation between those areas where there's academic involvement and outcomes. Outcomes are better in hospitals or in areas where there's a teaching hospital and where there are clinical academics. So it's better for your patients. So that is my talk. I want to thank you very much for taking the time to listen to me. And this is just to acknowledge the many, many, many people who've helped contribute to the research that I've done over many years here in Scotland and in America.
Professor Bashir, just unmute yourself, please.
Actually, I lost the sorry, I lost the keys for the voice and for the video. Sorry about that. I don't know where it is exactly. Yes, here we go. Now I got it back. Yeah, I think there is some sort of lagging, but thank you very much, Prof. Rory, for your amazing talk. It's very rich, it's very interesting. We enjoyed it so much. Now we open the floor for the questions, and we do have two questions. Interestingly, one of them is about research, and the second one is more of, scientific or clinical kind of question. The first question, it is about, a lady, her name is Amani, and she asking us about, some of the challenges that facing some of the clinicians when they doing research. And this is related to the fact that, for example, if you want to apply for ethical approval and if you want to, proceed any sort of clinical studies. Now, in UAE, we do have different committees to do so. So, and we do have different institute with different kind of committees to give this approval. So what, I can maybe just being here and aware of the process in general, I can, answer part of the question by saying that it depends, Amani, it depends on where you are, where you're practicing, and where you want to do your research. If it's, for example, for Dubai, there is committees in Dubai, in DHA, you can approach them. They do have a clear pathway that they can help you where to start and how to apply for that and to get the approval. The second, and also I should say that part of our initiative is as we're going into the training and into the support system, we will help all the people who are kind of having some challenges when it comes to applying for ethical approval and where is the right place for everyone to do so. Also, we do have, and I should say that we do have in the coming webinars, we do have one of the directors of the ethical committees in our university who will be talking about the procedure and about the process for applying for that. A second question was about a lady who's asking you, Rory, about she noticed that there is a variation of presenting certain symptoms when it comes to hypoglycemia. So she's saying that some patients present symptoms of hypoglycemia at 70 milligram, for example, where others are higher or lower. So can you explain that in any way? And I'm trying
Yeah,
to fix
absolutely.
my system. Yeah.
Absolutely. So, I mean, I think that relates to the point that I was saying earlier on, and that is that there's no single response. People vary, and that's why you have to study populations to look at mean responses. People will, this system is very, very flexible, how your body senses glucose, and it can change both dependent upon the prevailing levels of glucose levels. So if you've had high control, you'll often can't regulate a high glucose level. If you've had recurrent hypoglycemia, it's at a lower glucose level. But it also depends on other things. If people, for instance, are focused on doing the crossword or are doing some written work, they're going to suddenly notice neuroglycopenic symptoms, which they might not have noticed if they were out doing a run, and they notice different symptoms. And I think these things are all very context dependent. And also there is the final thing, which I think people forget about, is that symptoms are a perception, and perception is a very individual thing. It is not the same as a biological response. And I think we're inclined too often to think it's all about biology. And I've had people who you make hypoglycemic, who are obviously hypoglycemic, who cognition is impaired, and they'll say to you, "Oh, doctor, I'm not hypoglycemic at all. I feel perfectly well." And there is an inability to recognize that their symptoms are low. Others, of course, feel they're hypoglycemic at very high levels, even when they're not. So it's a really challenging area to do. And I've just always accepted that people are very, very different. And I explain to them that things can change all the time, that they need to confirm to recognize that they are hypoglycemic, and then try and teach them to manage it. So the explanation is that there are many, many different variables that can actually contribute to it, but it's really well recognized. And I think, you're right to pick up on it. If I were to say as well, Professor Bashir, very broadly, I mean, I feel your colleague's pain about how do you start research, because people can feel very isolated. And I think it's really important that you have things like the C.R.E.A.T.E network, because you can then offer people, because getting networks, seeking advice, sometimes being able to build systems to support research, like clinical trial centers, where you can offer training, it's absolutely crucial, because otherwise it's daunting. You look at all the paperwork, we've got terribly bureaucratic, you think how hard it is to get started, and just don't do it alone, seek that advice.
Yeah, thank you. Thank you very much for that, Rory. While we're waiting, waiting for more questions, I just want to ask you a question for, like what it takes for a clinician to be a scientist. So is it the passion and the interest, or is it the availability of resources, or is it, it has to be mandate? Like for all clinicians to be promoted, for example, or to be, like to go to the second stage to do research. What do you think, what it takes most for a person to be clinician?
I think that medicine is a broad church. And I think if you look at that community of practice, you're going to have people who are full-time clinical, and you're going to have some people who are part and part, and then some people who do lots and lots of research and others that don't. And I think it's a good thing. I think that's very, very healthy. I think in terms of those who go on to do research, part of it is passion, and you've got to be driven, because you've got to take risks. I resigned from a lifelong employment as an NHS consultant, and I took my family to America, where my job depended on my grants. If I didn't get grants, I had no salary. So you've got to be willing sometimes to take that risk. But you know what, I always figured out that I could be a doctor somewhere, so if it didn't work out, I'd just come back again. And you've got to have drive. You've absolutely got to have drive, because if you don't have that, if you're not resilient, it can be challenging. Now, that said, I think it can happen at any stage, and you shouldn't be put off. I mean, I was an NHS consultant, and I came back to do research. So you don't have to be someone who's decided at the age of 19 that you want to be a clinical academic. It can happen later on in life. I think it does have to be helped, and you have to be mentored, and you have to be supported, because it's not easy. But all of that is very variable, and you can pursue that research. If you've got a question you want to answer, then I think do it. I mean, life is too short. So if you've got the opportunity, I'd say as well, personally, I think it's a fantastic career. I know it's been challenging, but I love the variation in my career. I looked at my job in the NHS and I thought, I'm going to be doing the same thing for the next 40 years, and at some point that just becomes really tiring. And I really wanted to think about what could I do that was going to really make my day varied. And so I still love my clinical practice, but I enjoy the research as well.
Yeah, and I do have, there is a technical glitch. I can't see the question in here in the box. However, I have it here in my phone. So one of the questions we're asking you about is the training, high intensity interval training that you've done. And the question is where there are any specific recommendation for insulin dose adjustment or carbohydrate intake before or after high intensity training session to minimize hyperglycemia while still preventing hypoglycemia.
Yeah, really good point. Now, I think there are really good guidelines out there now. So I took part in a program through JDRF, now called Breakthrough Type 1. And we wrote an article for the Lancet where we gave a number of recommendations about how you manage carbohydrate and insulin with exercise. And as you all know, and as your colleagues will know, there are very, very different types of exercise. So if you undergo aerobic exercise, low intensity, long duration, that's generally going to, it's fat burning, but it's generally going to make your glucose level drop. And so you're advising people to reduce their insulin prior to that form of exercise, to take regular carbohydrate replacement. High intensity exercise, on the other hand, is likely to drive adrenaline release. And you may not know this, but adrenaline drives hepatic glucose reduction up to 10-fold, but muscle glucose uptake can only go up to 4-fold. So what high intensity exercise does is it drives your glucose up and it doesn't tend to go down. But then people make the mistake of taking insulin, and that creates then hypoglycemia afterwards, which is very, very dangerous. So I have a couple of general ways of doing this. I talk to people about the concept of insulin on board. And by that, I mean, let's say you exercise 2 to 3 hours after eating. Well, then generally you're just on a basal insulin. And so your insulin on board is low because your prandial insulin is mostly being used. You're short acting. So insulin on board, insulin's low, monitor your glucose. If you're doing any less than 45 minutes of exercise, you're going to be fine. It's not really going to be a problem. If you're going to do more than 45 minutes of exercise, you take 15 to 30 grams every 30 hours. Okay. Yeah. And you think about reducing your insulin for the meal afterwards. If you're doing exercise and you know you're doing exercise within 2 hours of eating, well, then you've got lots of insulin, you've got high insulin on board. So then you need to absolutely reduce your insulin before you exercise. And you can reduce that from up to 75%, depending on how much exercise you're doing, and you continue to monitor. The other thing is to deal with is that after the high intense exercise, when your glucose levels go high, you don't take insulin. What has been shown lots and lots of times, if you then do 5 minutes, 10 minutes walking, activate the C.R.E.A.T.E cycle, that glucose level will drop and you will not become hypoglycemic. And your hyperglycemia will correct itself. So it's that low intensity exercise after the high intensity, 10 minutes on a bicycle, gentle pace, or walking will actually bring that glucose level down.
Yeah, thank you very much, Rory. And we have a follow-up question regarding the same study from Zainab Al-Abadla. She's a diabetes educator. She wants your point of view regarding the findings and how is it possible to be generalized to a broader type 1 population, including those who are using hybrid closed loop system, or do you anticipate different outcomes in this setting or in this group of patients?
So that's a really good question because of how do you implement this type of therapy? And I have these questions all the time because, again, nobody's going to do high intensity exercise all the time, right? I mean, it's just not going to happen. And I think that there are two ways that I look at it. It is a short-term fix that people might do, a way perhaps to prevent people who developing impaired awareness. Longer term, the thoughts going through my head are simply that I think we are advising people with type 1 diabetes that exercise, different types of things are really important to their lifestyle, and they need to vary it up. And just doing the one form of exercise is not a good idea. You need to mix up your exercise. I mean, I'm a big believer in exercise. We have a condition where high glucose levels over time will damage vascular endothelium. And the biggest way to improve endothelial function is exercise. So, I mean, I would prescribe exercise for people with type 1. And my advice to them is just to vary it up. Sometimes it's high intensity, sometimes it's weight type resistance exercise, sometimes it's aerobic, longer duration exercise. That makes things challenging because there are different ways of managing insulin and carbohydrate, all of those, but there are very simple lessons that can be learned. And over time, people learn how to do it. In the longer term, could you talk about combinations of sauna, combinations of high intensity exercise? I don't know. I think that what it's telling us again, and this is hypothetical, is that the human body is there to be challenged, and that our systems need to be challenged regularly in order to stay responsive. So if we become inert, if we're not exercising, if our bodies are sitting in the one temperature control all the time, and we're just taking insulin and we're just being exposed to hypoglycemia, it's not very healthy for us. We need to vary up our lives a lot more to be healthier in the longer term. And that's for all of us, actually, not just people with diabetes.
Mm-hmm. Yeah, thank you for that, Rory. Still, we have a very interesting question related to the clinical and scientific aspect, but I want just to ask a question related to what we're trying to do in this C.R.E.A.T.E initiative, and then I will go back to the other question. So for young clinician, what do you think, what they need to develop to be successful scientists and clinician later on in their career? So what sort of skills they need most when they start their journey as young clinician who want to involve in research later on?
All right. Okay. So I think there are some general skills, and there are then some specific skills. So the specific skills it depends on what you're going to research. And I think your main skill is always going to be your clinical knowledge and your clinical practice. But within that, you need to have an area in which you are expert. So in my case, it was on mechanism, right? Because much of my early work was around mechanism. For others, it's on health data science. And so their expertise is in how do you handle big data and some of the statistics that comes with that. So that's their area. So you need to have an area in which you are developing a special skill set. I think then to succeed, you've got to have a few things. I mean, you've got to learn about critical thinking. You have to be able to write. All right. It's critical that you can write, and you only do that by practice. You do that by not just by writing papers and writing grants. You do that by reviewing papers and reviewing grants, because that teaches you what's a good paper, what's a good grant. So you must try and do all that work. It's a lot of work, but it's really valuable for you. You need to build your networks. This is absolutely the era of team science. So individuals in their lab doing it on their own doesn't work anymore. So it's trying to make contacts with people who can bring in interdisciplinary, different skill sets that can help you do what you want to do. And then I think at an early career stage, it's really good to get a mentor, someone more senior who's been through it before, who can advise you and talk to you more generally about the challenges that you might face and how you might overcome them.
Mm-hmm. Yeah, thank you very much for that. And by the way, all the questions that we're receiving, everyone is, saying thank you very much for this very interesting talk. There is a very interesting question or comment, a person who highlighted how much he likes the concept of this habituation and how you correlated with the counter-regulatory response. But there is, like a point that he wants to know your point of view about it. So we always talk about down regulation of the of this response. He's saying that, do you think there is any correlation between overactivation of the counter-regulatory mechanisms when it comes to the chronic hyperglycemia patients?
Yeah, I think it's different, probably different systems, but possibly yes. And so if you look at people with high glucose over time, then things like cortisol levels will be higher, and you move into this chronically inflamed condition with activation of those counter-regulatory responses. And I think that contributes to the downside of chronic hyperglycemia. So in that sense, yes, I do think that the systems are to some degree upregulated.
All right. Another question, of course, we just on time, I'll try just to add a couple of questions. We're still receiving many questions, but unfortunately, due to the time, we can't accommodate all of them. But there is a question about, patients who have autonomic neuropathy and how this will affect their symptoms when it comes to hypoglycemia. Do you think we should have a different guidelines when it comes to diagnosis and preventive measures in this group of patients?
No, actually, I don't, because I think that this cohort have been studied before, and in fact, the symptomatic response to hypoglycemia is the same in people with autonomic neuropathy who don't. I think you just deal with a group of individuals who've got multiple complications of very long disease. So, in response to your questions, I don't actually think it does massively affect the symptomatic response, or at least not so far as the research has been able to show.
All right. And I think this is the final question. We still have many questions coming, but maybe, like in a different occasion, we can exchange that with you and we get your point of view. A very interesting question about somebody asking how to avoid rejection in a high impact journals. Your recommendation for people who want to publish in high impact journals and they always get rejections.
Yeah, so well, there's a couple of things. First of all, I think try and ignore impact factor. I mean, a lot of really high impact journals like Nature, and I don't even read them anymore because half of the stuff in them is just, you can't tell. And so sometimes I think citation is more of an evidence of influence than impact factor. Diabet Lojo, I think, is a great journal. I think there is bias out there. There always has been. There are groups that are really well known who sail into journals that are high impact. There are groups who are less well known who find it more challenging. I think you've got to persist. I think you've got to look at the reviewers' comments. If they're actually not very good, then challenge them and go back. If, on the other hand, they're really valuable, take it on board, change your paper, go to another journal and get it out. There's lots of journals out there now. And I think it's important to try and do this. So don't give up. Keep going. Learn from those responses. I have been rejected so many times, it's not true, from different journals in my days, and it happens to all of us. And sometimes, as I said, sometimes I think, actually, no, that review was rubbish. It was wrong. So I'll go back to the editor and tell them. They might not agree with me, but at least I've said it. But at other times, I'll take it, rewrite it, rethink it, maybe do some more work, and then I'll go to another journal.
Yeah, definitely. I totally agree. By working in the comments from the reviewers, it is a learning process and you keep improving as you go through the way. Thank you very much, Prof. Rory, for your time, for your amazing presentation. We learned a lot and we enjoyed it. Thank you very much for spending this time with us. We learned a lot, and hopefully we'll see you again in the coming webinars. We really appreciate your presence.
My pleasure. Thank you very much for inviting me.
Thank you. Thank you. Thank you, everyone else, for being here. It's Thursday night, but being with us, sparing time to be with us. Thank you very much for being here. And as we said before, it is, like all the webinars, the same accredited. And to make sure that you're getting that, you have to fill in the evaluation form that you will receive after our session today. And everything that we've done today, it is available on demand on the EDES Institute website and also in EDES YouTube channel, which is the Endocrine Diabetes Society. And please make sure that you are visiting this website because we have more exciting presentations coming. Our next meeting, it will be on the 25th of September. We have another excellent clinician who are a scientist and doing a great job when it comes to impactful research. It will be about turning clinical challenges into research opportunities. I think somebody opened the mic all of a sudden. So this will be delivered by Prof. Salah Abusnana on Thursday, 25th of September. Also, we do have more of very interesting webinars will be happening in the coming months and in October as well. Please stay tuned and pay attention to our announcement. Another couple of notes, it's related to another important forum that's happening and it's started and sustained by Emirati Diabetes and Endocrine Society, which is Young Leaders Forum. We have a very exciting activities coming, so please be with us in all of that. And we do have the big announcement. And Prof. Rory, please, we want you to be with us in EDIC 2026. This will be the 16th version of this conference. It is one of the prestigious, very popular meetings in UAE. We'd be very happy to have you. That will happen on the 6th to the 8th of February 2026. I think I'm done with everything. A huge thank you for every single person who came today to be with us. Have a great evening and a great weekend, and we'll see you next time. Thank you.
Many thanks.